Today I will be covering one of the most important biological processes, photosynthesis. Most people have a general understanding of photosynthesis, where plants take in carbon dioxide, water, and a little bit of sunlight in order to produce carbohydrates and oxygen as a byproduct. This is a very simple notation of photosynthesis, but it is not incorrect. Let us, however, delve deeper into the phenomenon of photosynthesis. The process of photosynthesis can be separated into two components. We have the light reactions and we have the subsequent dark reactions. We will first take a closer look at the light reactions. The names light and dark reactions are names that scientists have determined are pretty dumb names, since dark reactions still take place in the light. So rather than calling it the dark cycle, we will call it the Calvin cycle. As for the light cycle, it was more appropriately named the light-dependent reactions. As mentioned earlier, we will look at the light-dependent reactions first. In order for a light-dependent reaction to take place, the plant needs, well, obviously light, but also water, and it will result with ATP, NADPH, and oxygen as a byproduct. Remember this reaction, or more specifically the products of this reaction, as they will drive the explanations in this video. Of these three products, we will first focus on the production of ATP, and will follow with NADPH and oxygen. What you are looking at now is a crude attempt of a zoom in on a thylakoid membrane. The thylakoid is the weird disc thing inside the chloroplast. The membrane is a phospholipid bilayer. This is photosystem 2, and this over here is photosystem 1. This is, as it's labeled, ATP synthase. Our journey begins at photosystem 2, where light enters into the system. Within photosystem 2 is something called P680, which has an electron waiting for the light. The light will excite the electron of P680. These excited electrons are in a higher energy state. Oops, I forgot to mention some crucial parts of this diagram. The inside is called the thylakoid lumen, and the outside is called the stroma. Now, this electron will be transferred from one molecule to another, and it is going to go to lower energy states. Some of the energy that is released during this process is used to pump hydrogen ions across the membrane, creating a concentration gradient between the stroma and the thylakoid lumen. Please ignore the fact that I have placed the hydrogen gradient upside down in the stroma. This hydrogen gradient can be used to fuel the production of ATP by ATP synthase. An ADP and an inorganic phosphate nearby will be jammed together by the movement of hydrogen ions. The ADP and the inorganic phosphate will come together to form ATP as a product. This flow of hydrogen ions through ATP synthase from the thylakoid lumen into the stroma is called chemiosmosis. Once this electron gets donated and goes through the cycle, you might be wondering, well, how does this electron get replaced? Well, the electron came from this P680 molecule. When this molecule loses an electron, it becomes a very strong oxidizing agent. So strong, in fact, that it will oxidize the nearby water molecules. Essentially, it oxidizes the oxygen in the water. Once the P680 grabs an electron from the water essentially falls apart, leaving the oxygen and the hydrogen ions. The hydrogen ions contribute to the concentration gradient, and this is also where we get the oxygen byproduct. Here we have 1/2 of an O2, so if you do this twice, you're going to have a molecular oxygen. So far, we have covered the ATP and the O2, but what about the NADPH? We will talk about that now. As I rebuild the membrane and question why I even moved it away to begin with, allow me to explain a bit about the photosystems. Photosystems are combinations of proteins and molecules, and also have chlorophyll and variations of chlorophyll, and pigment molecules that are very responsive to light, and have electrons that can get excited by the light. With our membrane put back together, let's get started. Similar to what occurs during the production of ATP, light enters the photosystem. Only this time it enters photosystem 1, home to a slightly different molecule, P700. Once again, the electron gets excited, leaving P700 as a very strong oxidizing agent. To replace the electron on P700, sometimes the electron from photosystem 2 will actually come over and reduce P700. Now, the thing that makes photosystem 1 different is where the electron ends up. As it goes to a lower energy level, it goes from one molecule to another, and it can be used to reduce NADP+ and make it become NADPH. We have now discussed how ATP, NADPH, and oxygen are produced in the light-dependent reactions. Now, let's shift over to our second of the two reactions involved in photosynthesis, the Calvin cycle. The Calvin cycle takes the ATP and the NADPH from the light-dependent reactions in an outside carbon dioxide and forms that into sugar. Once again, remember this reaction, as it will be what I refer back to a lot of times. In plants, carbon dioxide enters the interior of a leaf via pores called stomata and diffuses into the stroma of the chloroplast, the site of the Calvin cycle reactions, where sugar is synthesized. These reactions are also called the light-independent reactions because they are not driven directly by light. In the Calvin cycle, carbon atoms from the CO2 are incorporated into organic molecules and used to build 3 carbon sugars. This process is fueled by ATP and NADPH, which were produced from the light-dependent reactions. Unlike the light reactions, which take place in the thylakoid membrane, the reactions of the Calvin cycle take place in the stroma, the inner space of the chloroplasts. The Calvin cycle reactions can be divided into 3 main stages: carbon fixation, reduction, and regeneration of the starting molecule. I will go through each stage, starting with carbon fixation. Carbon fixation starts with the CO2 molecule, which combines with the 5-carbon acceptor molecule, ribulose 1,5-biphosphate, or simply RUBP. This step makes a 6-carbon compound that splits into 2 molecules of a 3-carbon compound, 3-phosphoglyceric acid, or 3-PGA. The next step is reduction. ATP and NADPH are used to convert the 3-PGA molecules into molecules of a 3-carbon sugar, glyceraldehyde 3-phosphate, or G3P. This stage gets its name because NADPH donates electrons to, or reduces, a 3-carbon intermediate to make G3P, resulting in an NADP+ at the end of this stage. The final step is regeneration. Some G3P molecules go to make glucose, while others must be recycled to regenerate the RUBP acceptor, the crucial part in the first step of carbon fixation. Regeneration requires ATP and involves a complex network of reactions, and since I don't see where I need to explain that on the rubric, I'm not going to. In order for one G3P to exit the cycle and go towards glucose synthesis, 3 CO2 molecules must enter the cycle, providing 3 new atoms of fixed carbon. When 3 CO2 molecules enter the cycle, 6 G3P molecules are made. 1 leaves the cycle and is used to make glucose, while the other 5 must be recycled to regenerate the 3 molecules of the RUBP acceptor.